Okay, thanks for the organizers for this opportunity to speak to you all this morning. So yes, as Stefan pointed out, I'm going to talk about some computational approaches that we're making, and I'm splitting my talk into two parts, one on data visualization and then another on more chemistry approaches. So if I can just the correct one. Bear with me, sorry. Should I click one of these? Oh, this one worked. Yeah. Okay, great. Okay, thank you. Okay, so our goals in terms of trying to visualize the epigenomic data, of course, so clearly we want to complement an automated computation, and I think humans are still pretty good at identifying patterns and features in data that computers aren't. So human interpretation is still an important aspect of what we do. And it helps, of course, make the data accessible to a much larger group of people. Now, Ting gave a talk yesterday about his great epigenome browser, and these approaches show the epigenomes across a functional unit, a gene, and so we can quickly understand what's happening at the promoter and in the introns and across the exons and across the length of that gene. So obviously, Ting was looking after that, but what we wanted to do is to look at the analysis in a different dimension. And that dimension would be across specific features. So instead of looking across an entire gene, the kind of questions that we wanted to ask is, what if we looked across just the promoter regions, or just intron splice sites, or just the first exon? What are the epigenomic patterns across those features? And we think that would be insightful to understand the number of different patterns, for instance, that exist across promoters in a particular cellular state. So again, this is what Ting was doing. And so what we developed was a clustering approach to identify the different patterns of epigenomic modification across different features. And we call this software Spark. And what I'm going to discuss in this part of the talk is primarily the work of Dr. Sidney Nielsen. Okay. So what we'd want to do, for instance, in this region here, is that we've actually got 3 genes, and they each have a promoter, and you can see that there's kind of different marks collating or collecting across those particular features. And so if we can pull just those regions out down here and kind of normalize the signal and also, of course, correct for the strandedness, we come up with different patterns across those different promoters. And then, of course, if we take all the promoters across the entire genome, we've got a whole bunch of different kind of matrices that reflect each promoter. And then our goal would be to do a fairly straightforward now clustering of those and identify the different kinds of promoter marks or epigenomes, the collation of marks across each of those promoters. So what we can see here, for instance, if we just take one cluster, is that what happens is that there's one very clear set of promoters that for instance, have accrued H3K4ME3 and H3K9AC, and the other collection of promoters haven't to any great degree. And if we look at the RNA-seq, we have a readout from the cell. So this is the only cluster that's actually associated with transcription from those promoters. So we can quite quickly see what are the active promoters, what are the inactive promoters, and what are the different kinds of patterns that we see across the promoters within that particular cell type. And of course, we want to dive down, so the software allows you then to say, well, what are the genes within that cluster? And although there may be a lot, we can still actually go and look at the pattern across any individual gene at that point. And of course, we can then go back and look at what the patterns were, the precise kind of bed files that were for that gene if one wanted to. So it's important to discover the kind of patterns of promoters and then be able to drill down to the actual gene feature that we're looking at. Do these clusters have functional meaning? So if we see a pattern across a promoter that we haven't actually seen before or we don't think is that common, an obvious question would be, does it correlate with any particular functionality in that cell? And obviously there's lots of software that kind of uses ontologies to do that, and so we've simply used the gene ontologies that are available to say, what, for instance, the genes that have this specific pattern here, is there something about them? They're marked with H3K4ME3, but not H3K9 acetylation so much. What do they mean? And so we can essentially plug into David and send that query off to the David platform. And so that can give us some insight. And there's a number of kind of file formats that the software will accept. There's also a website, and there's also some videos that allow people to learn about how the software can be used. We also published it on Genome Research, and the only reason why I show this is kind of an artist stylization of the clusters of epigenomic patterning across the different promoters. And then just as a kind of use case example, how we've used this, so we collaborated with Peggy Farnham in this case. And so in Drosophila, for instance, there was a fairly well-known kind of molecular biology interaction where the FORC protein is basically recruiting PRC2, which does the trimethylation of H3K27, which then allows PRC to come along and actually instigate then the transcriptional repression. And one question was, does the same model actually hold in human cells, and in this case, the human stem cells? And so you can see that YY1 and SUS12 are actually the human equivalents. And so we would expect, for instance, YY1 to co-localize with SUS12 and actually the H3K27ME3 mark. And so when we do this experiment, what we actually see is that actually, if I just go to the next slide, the YY1, there's actually no real strong association with YY1 accumulating in any of these promoter types with basically H3K27ME3. So that actually showed that these cells were actually behaving in a very different way to what we actually expected. But what the software was also able to allow us to see is that CTBP2 actually co-localizes with YY1. And what we can appreciate too, actually, from this is that CTBP2 actually co-localizes with YY1 in a mutually exclusive pattern, right, with SUS12. So if ever there's basically CTBP2, we really don't see SUS12 showing up. So that was actually a really interesting, quick observation that actually led to a publication where we were able to see the behaviors across these promoters and appreciate some biology actually fairly quickly. Okay, so what we're able to show is, of course, that only a minority of the YY binding events correlate with H3K27. This mutually exclusive complex forms with SUS12 and CTBP2. And so we were able to identify this kind of novel co-localization of these transcription factors across active transcriptional start sites. We've kind of moved on with that software a little bit too, and this is in collaboration with Torsten Moller and Hamid Younsi at SFU, where we've taken the same approach, but actually to look much more in detail at the pattern of the marks across these promoters. So they could be actually quite insightful. I'm not going to really go through what we're trying to do, but instead of just kind of blocks, these are the kind of patterns of the marks that accrue across a certain feature. Or we can kind of blow up here, and we can see, for instance, that across a different feature, the relative position, there are some differences in the way that the marks accrue, or there's some similarities in the way that the marks accrue. And again, that could be something that's very biologically informative. So we did publish this approach, and actually there's now a website with that particular software now available as well. Okay. So in the second part of my talk, I mean, the question that we wanted to ask too is, we're learning about the epigenomes, and clearly my interest at a cancer agency is how do the epigenomes interact with oncogenesis and how they're involved in cancer as a disease. But at the same time, how can we kind of use these tools as ways to mitigate the disease and have compounds that could possibly interfere with the epigenomic patterning. So aberrant expression, aberrant epigenetic modification is a key feature actually in a large number of human cancers, and that's been known for a while, but I think we're appreciating the number more precisely nowadays and the actual genes that are involved in particular cancers. So in the cancer that I'll be talking about, we're using kind of lymphoma as a model, and we've been working on that for a number of years. And in that disease, a common feature would be that we've got hyperactive repression across many tumor types, at least in lymphoma. And what the repression does is stops terminal differentiation pathways and keeps these cancer cells in these kind of proliferative stem cell-like states. And we also see mutations in genes involved in activatory marks, because instead of repressing differentiation pathways, what these mutations are doing is preventing the activation of differentiation pathways. So they're actually doing the same thing, just in different ways. They're keeping these cells in proliferative stem cell-like states. And so we published a couple of papers really in lymphoma, identifying MLL2 as a very recurrently mutated protein in that disease, and EZH2 as well as an epigenetic modifier that actually has a gain-of-function a gain-of-function allele that's actually very common in this disease. And we can see this when we look at these genomes. What we see across maybe, I think, about 40 genomes, possibly more in this set, is that EZH2 is only essentially mutated at this spot here, at this position here, and that's a gain-of-function allele. I'm not going to go into how that allele works, but it's now pretty well characterized. And ML2 seems to accrue mutations across its entire length, and that's very consistent with loss of function of ML2. So how do we exploit this? Well, EZH2 and ML2, these are histone code writers. EZH2 is gain of function, ML2 is a loss of function. And we know how these can prevent the activation of these kind of differentiation pathways. What we want to do is to say, well, these proteins write these marks, but can we actually interfere with the proteins that read those marks, right? And in that case, it'd be CBX2 is reading the mark of EZH2, and BPTF is reading the mark of ML2. And so that was kind of our challenge, is can we develop a kind of therapeutic strategy using those two genes as targets. So essentially what we're saying in this disease, that there's kind of two processes that can happen that effectively do the same thing. And so what we're saying is that if in the tumor MLL2, the writer is knocked out, right, to mitigate that, why don't we modulate the activity of the reader BPTF to compensate? Likewise, if the EZH2, the repressive writing mark EZH2 is upregulated, why don't we kind of chemically modulate CBX2 as a reader to compensate for that hyperactivity? And so what we were able to do is we developed, or we computationally kind of screened or predicted a number of compounds that could do exactly that. So CBX2, for instance, we have a compound here in turquoise, and this is actually in purple. This is the histone tail coming in. This is actually the monomethylated tail, and the idea is that it interferes with the monomethylated tail in this case. Very similar for BPTF. We have a compound that we can get into the active pocket in BPTF and interact with the histone tail, in this case the K4ME1, the monomethylated form. And so the idea would be that by using these compounds in this way, we can modulate the substrate specificity of these epigenetic readers. In fact, we're trying to reprogram, right, the epigenetic program. So CBX2 is kind of a simple concept. You've got a hyperactive process, so actually a simple inhibitor would suffice. So that actually, I'm not going to talk about that because that's fairly straightforward in how you do that. The kind of more interesting thing is, how do you compensate for the loss of function of a protein in a tumor? How do you get around the fact that a tumor suppressor has been lost? So what happens in the cell, if you just ignore these accoutrements at this point, BPTF basically binds the trimethylated form of H3K4 in this way. And all our understanding is that basically it has little affinity for the monomethylated form, and in fact little affinity for the dimethylated form as well. And so what we wanted to do is to identify a compound that would basically, presumably, prevent this from happening, although that may not be that important, but a compound that would allow this to happen. So a compound that would go in and actually allow the monomethylated form to bind in the same way that the trimethylated form would actually bind. And you might notice then that these kind of unnatural accoutrements next to it give us a way of actually measuring that interaction from like a pull-down gel-based assay. And so these are the kind of the results that we have from a couple of compounds. I show some gels just up here for illustration, but these are the results actually from a number of different replicates. And we can see that the two compounds that we've identified so far basically modify the behavior of BPTF in different ways. So this one here, in the presence of the compound, actually, instead of binding to ME3, actually now gives a much stronger binding to the non-methylated form and some binding to the monomethylated form, and seems to select against binding now of any higher methylated numbers. And you might expect that, seeing that we've shoved a compound into that pocket, that there's less space. We see a similar thing with the other compound, but what's nice is, I think, as we characterize these further, is that there's a very different behavior. So this one really prevents ME3 now from being read by BPTF, and also ME2, but actually shows a nice kind of consistent liking in stabilizing ME1, but doesn't bind ME0 in contrast to this compound. So it's kind of nice that we have some different behaviors in which to play with. Okay. So when we test these, and actually we did this experiment before we spent all the time doing the gel pull downs, but what you want to see is that genes or cancers that are mutated for ML2 should be really sensitive to these compounds, because these are restoring the lack of function of ML2. And so what we can see is that the genes that are wild type in the presence of these compounds are basically unaffected by this compound, which is nice, because that's what we would predict. In red are the cells which are actually they actually have the ML2 mutation. And we can see that in red, 1, 2, and actually we don't even see a signal for this one, that the ML2 cells are actually very, very sensitive now to these compounds, again, which is nice because we targeted them against those cells. And then we have these cells here, which actually, if this is yeah, actually, because this color came out confusing, but these green cells here are actually a double mutant. So they should be sensitive somewhat to this compound because they bear the ML2 mutation, but they also have this enhanced gain of function repression. So it's kind of, if we can get around this, it's not clear that we're getting around this as well. So we get an intermediate response, which makes some sense as well. So that's basically, I mean, we've done some mouse work and it shows effects too, but I'm not showing that. But basically, in the time I got left, I mean, I'm just going to show really what our idea is, again, in terms of epigenetic reprogramming or kind of remodeling using chemical moieties, right? So I mean, essentially, if we have on this slide, for instance, in a normal cell with functioning ML2, in the desired regions, we should see a nice binding of BPTF across the trimethylated forms, and presumably that there'll be other of lower methylated forms hanging around as well. And in the cancer, one of the reasons why it's a cancer is because it's basically lacking these trimethylations, and BPTF isn't able to bind. And so really, just in summary of what we've been talking about, the idea is that if we can give this compound, for instance, this should now start to make this cell look kind of more wild type. And again, if we add another compound, it should make it even more wild type. And so that's the goal, is to make that tumor cell more wild type, in which case it basically should just terminally differentiate, and certainly hopefully wouldn't be a clinical problem. Yeah, and of course we don't have anything that would bind this at this point. So anyway, I think my time is up, and I'll just thank the large number of people that we've worked with, both at the GSC and obviously that we're collaborating as well. And these are the people at the Genome Science Center. Obviously, we worked a lot with Joe Costello's lab and people at UC Davis and David Hausler, where Ting was, and of course now Ting in Washington University. So again, I'd like to thank everyone for your attention.
Thank you, Steve. Questions?
Steve, with your compounds, what sort of selectivity have you done? I mean, what are there likely to be any downsides of compounds that are binding monomethylated chromatin, for example?
Oh, okay. Yeah, I mean, it's interesting. I mean, we can get these compounds, the H-stack inhibitors you'd think would be totally poisonous, but they do have some therapeutic appeal. In oncology, that's not such a problem because these are fairly desperate diseases, and toxicity is acceptable as part of a treatment. It's just the therapeutic window that's important. I alluded to the mouse work. We've done, the maximum tolerated dosage tests. So, we know that we can get them into mice at actually a fairly good level. They don't keel over. We know that we can get them the compound into at levels that actually slow down or mitigate the growth of the tumor. These models grow, we can only do the experiment for about 9 days because they grow so fast, so we want to probably get a better model. But these are really unoptimized compounds, right? They're from the initial screen. I think the idea is to do some medicinal chemistry to get the specificity even higher. But anyway, that's as far as we've got.
See no further question. Yeah, we have one more.
Steve, MLL and EZH2 mutations likely are secondary or tertiary in follicular lymphomas to 1418 and CREP-P mutations. So do you expect an effect on those bystander or secondary clonal evoluting cells which do not have those mutations?
So I'm not sure. I mean, it's not clear that EZH2, for instance, is what you say is a secondary mutation. It's very, very recurrent, right, as an example. So it probably means that those tumors really need that mutation to become clinically problematic tumors. Both Glaxo, SmithKline, and Epizyme, I think it's Epizyme, they've both got inhibitors to EZH2, and those show, again, targeting EZH2, they're able to show, for instance, that they can slow down tumor growth in a mouse model. So I think it still means that they're relevant targets at this point. Yeah.
Thank you, Steve. We better